Okay. Sige, allow me to share my screen. Before we start, sandali, bakit nag-feedback? Ayan, okay. Before we start, I am expecting participation from the group, okay? Since this will become interactive. Wait lang, paano kaya mag-mute nito? Mute. Okay ba, everyone? Can I expect na mag-participate kayo? Yes or no?
Yes po.
Okay, mag-participate lahat ha. So nakikita ko kayo may other screen. Sige, let us start. Kita na class, everything yung screen ko.
Opo.
Okay, sige. So our topic for tonight is Fisheries, Biotechnology. So supposedly before hindi to sinasama sa mga online discussion. However, sabi ni Sir Rico Toong last time, yung ating top 2 sa board exam, bago na yung ating set of examiners, so better na i-add natin si Fisheries Biotechnology. It's because we also have subjects about fish breeding and genetics. So this, mute na lang po nung inyong mga mic. Okay, sige. Let's start. Okay, number 1, first question. Stores the information needed to build and control the cell. So what is the answer? Pwede kayong mag-reply dito sa chat, everyone, para makita ko kayo, yung mga sagot ninyo. Question number 1, stores the information needed to build and control the cell. A, DNA or deoxyribonucleic acid. B, ribonucleic acid. C, chromosomes. Okay, so what is your answer? Okay, sandali ha. Meron kasing okay na, nakamute na. So, yung mga answer na nakita ko dito, merong sumagot ng letter C, genes. Pero most of you answered letter A. How about the others? Anong sagot ninyo? Merong sumagot ng letter B. Okay, so the correct answer is DNA, letter A. DNA or deoxyribonucleic acid. So the explanation here, okay, so merong sumagot ng letter ng genes. However, yung question doon kanina is what stores the information? So it is DNA. DNA, deoxyribonucleic acid, it stores the information needed to build and control the cell. And we also have RNA, ribonucleic acid. Remember that it does not serve as the hereditary information in most cells. However, for some viral viruses, this will serve as their genetic material. Okay? And dito, merong sumagot na genes, Okay, so genes, it is the basic physical and functional unit of heredity. Basically, genes are made up of DNA. Now remember that your DNA can be seen in the nucleus of the cell, and then in the nucleus of the cell, you can see there the chromosomes, and then when you unwind the chromosome, ito na siya yung DNA. And then there in your DNA, makikita doon si gene or the sequence of yung tinatawag last time. If you remember yung lecture ni Sir Marnel Alberio, itong TCAG. Okay, so these are the 4 bases: thymine, cytosine, guanine, and adenine. Okay? So that's it. Now you know the difference among these terms. So next, okay, question number 2. The set of genes being expressed at any given point in time determines the cell's activities and its observable characteristics. So what is the answer? A, genotype, B, phenotype, C, both A and B, D, none of the above. So what is your answer? It is the set of genes being expressed. Okay, how about the others? Anong sagot ninyo? So meron nag-answer ng A, meron nag-answer ng C, both A and B, meron naman nag-answer ng letter B. Okay, so let's wait for the answer of others. So most of you answered letter C and B. C and B. So genotype, phenotype. Okay, so yung pinaka-dominant answer is letter C. Okay, so what is the correct answer? It is phenotype. Phenotype. Class, tandaan ninyo ha. Pag sinabing set of genes lang, it is the genotype. However, meron ditong term na expressed. Okay, expressed siya. So it is the phenotype. Parang ito, for example, you have this set of genes na that will express na kulot yung buhok mo. So yung sequence na yun, for example, ACTGGGCA, ganon. Now, yung kulot na na-express sa'yo, that is the phenotype. Okay? So, here is genotype versus phenotype. Genotype means the full collection of genes that a cell contains within its genome, whereas phenotype is the set of genes being expressed. Now, para ma-distinguish nyo yung dalawang ito, okay? Genotype, gene, full collection of genes. Pag nakita nyo naman yung term na being expressed, it is phenotype. Phenotype, okay? So next question. Question number 3. The application of molecular biological methods to marine and freshwater organisms. So what is your answer? A, genetic engineering, B, genetics, C, recombinant DNA, D, fisheries biotechnology. Okay, how about the others? Hinintay ko yung sagot ninyo. Okay, lahat sumagot ng letter D. Okay, how about the others? Letter D yung mga sagot, mostly ng mga students. Okay, so very good. The correct answer is fisheries biotechnology. Now, na-observe nyo kanina sa first 2 questions natin, yung iba ditong mga terms, after ng question is dinefine ko siya. So that is my technique when I was reviewing for the board exam. For example, merong A, B, C, D. Titingnan ko yung definition ng kada isa para matandaan ko na hindi na yung parang babalikan ko na naman ulit, ano yung meaning nito. So dapat along the way, while you are studying these different terms, look at their definition. Okay, so very good. Lahat sumagot ng letter D, which is fisheries biotechnology. Again, tingnan nyo yung root word dito, application of molecular biological methods. So that is biotechnology. And it is applied to marine and freshwater organisms, so that is fisheries. So kinumbine siya, that is fisheries biotechnology. Okay. So when we say genetic engineering, basically that is manipulating the DNA to alter the organism's genotype so that a new phenotype will be expressed. And we all know dito, I know lahat kayo, meron kayong subject na genetics. It is the scientific study of genes and heredity. Okay? Recombinant DNA naman is a technology that uses enzymes to cut and paste together DNA sequences of interest. So basically, recombinant DNA is used always here in genetic engineering. It's because here in your recombinant DNA, you are cutting and pasting DNA sequences together. So this is just one tawag dito, in one umbrella of genetic engineering. So baka ma-confuse kayo ha, fisheries or blue biotechnology. So under the umbrella of biotechnology, may tinatawag tayong blue biotechnology, green biotechnology, red biotechnology, and white biotechnology. So for blue technology, that is of course fisheries, marine aquatic resources. For green naman, obviously that is agriculture. For white, that is pharmaceutical. Red, that is industrial biotechnology. Okay? So very good. Question number 4. Which of the following is an application of fisheries biotechnology? Letter A, improved growth rate, FCR, or feed conversion ratio, disease resistance of fish. Letter B, improve tolerance to low water quality and cold tolerance. C, promote increase in fertility, reproduction, and promote harvestability. D, all of the above. So what is your answer? So may mga sumagot na dito na mga students. Yung sagot nila is letter D, all of the above. How about the others? Okay, so most of you answered, all of you pala answered letter D. So that is correct. Okay, letter D. So all of these mentioned here are the application of fisheries biotechnology. So basically, as you can observe here, it is for the improvement of our aquatic resources. Okay, look at this, fertility, reproduction, harvestability, FCR, disease resistance. So later on, matatouch natin itong disease resistance din na to, and then on how to improve the FCR and then the growth rate. So these are the effects of biotechnology in fisheries. So all of this, basahin nyo na lang to isa-isa, and then tandaan, pag may nakita kayong mga improved na term na yan, yun na yun. So itong nakikita yung dalawang isdang ito, mamaya matatouch natin ito. So this is the regular salmon, and this is the genetically modified salmon. So ang dami dito, improving fish quality and everything, especially in aquaculture. Next question, question number 5. A technique whereby ripe fish breeders are stimulated by pituitary hormone or any other synthetic hormone introduction to breed in captive condition. Letter A, monosex fish culture. B, induced breeding. C, hybridization. D, sex reversal. So may mga sumagot na dito ng letter B. How about the others? Okay, most of you answered letter B. Sige nga, magtatanong ako sa mga students na andito, bakit sa letter B yung sagot ninyo? Ano yung inyong merong sumagot ng letter D, which is sex reversal. Now, sa mga sumagot ng letter B, ano yung reason ninyo why you answered letter B? Ano ba yung term na hinahanap ninyo? Merong sumagot ng hybridization. Okay. So, tanungin ko si Honey Jean. Honey Jean? Are you there? Ano yung term na bakit mo nasabi na induced breeding yung answer? Ano yung term na nakita mo bakit ganun yung sagot mo? Hear from the question. Honey Jean? Okay. Ano si Honey Jean? Baka walang audio. Okay, sige. Rafael. Dinig pa ako, guys. Rafael, are you there? Okay, walang nasagot ng bakit walang nasagot ng mga students? Okay, sige, Mary Jane. Thank you.
With the use of synthetic hormones po, tapos it is bred in captivity.
Okay, ito yung term na nakita ni Mary Jane, synthetic hormone and breed in captive condition. So meron siyang dalawang nakitang words or phrases na nakapagsabi sa kanya na ito yung tamang sagot, induced breeding. Okay, sige Rafael. Okay, so the correct answer is very good. Induced breeding. So tama iyon. So class, remember to look on the sentence. Huwag tayong magmadali pag nagsagot tayo, doon tayong maghanap ng clue sa sentence mismo or sa question mismo. Kasi minsan, alam nyo, yung sinasabi ko, yung exam, common sense lang talaga yung sagot nakikita na mismo doon sa tanong. So it is induced breeding. So why? So induced breeding is a technique whereby ripe fish breeders are stimulated by pituitary hormone. Tandaan nyo to ha, pituitary hormone, of course, from the pituitary gland, which can be seen on the head of the fish. So, examples ng mga ginamitan nito ng induced breeding, ito, mga catfish. Okay? So, yung donor kasi this can be seen sa sa yung pituitary gland nakikita sa sa head ng isang organism, so you have to extract it. So the most widely used fish donor is Cyprinus carpio or our grass carp. Okay? So why pituitary gland yung ginagamit? It's because it secretes or it produces gonadotropins. Okay, like FSH or the follicle stimulating hormone and LH, luteinizing hormone. So FSH, tandaan ninyo, this is it causes growth and maturation of ovarian follicle in females and spermatogenesis in the testes of the males. And LH causes luteinization in females and promote the production of the testosterone in males. Natandaan nila ito itong dalawang hormones na to ha. FSH and then LH. And laging tandaan na galing ito sa pituitary gland and usual na hormone, most widely used donor pala is Cyprinus carpio or the carp. Okay, next, ito pala yung induced breeding techniques. Ito yung process. Now, so for an exam, minsan may mga lumabas na mga processes. So kunyari ito gagawin nilang A, itong process, removal of glands, tapos B, spawning, C, preservation of glands, D, ganon. So it's up to you kung paano nyo siya pagkakasunod-sunod sa tamang sequence. So dapat, ang daming mga methods na pinresent last time ng ating mga previous speakers. So dapat matandaan nyo ito lahat or at least maging familiar man lang. So these are the techniques. Number 1, of course, removal of glands. Ito yung sinasabi ko na removal of the pituitary gland from the head of the carp. Preservation of glands, usually yung ginagamit dito is acetone or alcohol. And then ito yung itsura ng prepared na gland extract. So this was pelletized. And then brooder selection, pipiliin if mature na ba yung brooder na gagamitin, and then i-inject siya intraperitoneally and then spawning. So yung egg and then yung milk ng both fish are tawag dito i-fertilize. So nakikita ninyo, this is the milk, MILT. Okay, so may sumagot kanina na monosex fish culture. So monosex fish culture class refers to the culture of either all male or all female population. So under the monosex fish culture, if you will read other articles pa, may ginagamit din dito mga synthetic hormones in order to change the sex of a fish, for example, from female to male. Okay, so pwede siyang all male progeny. Obvious naman, gagawin mong lalaki lahat, and all female progeny, gagawin mong babae lahat. So usually itong all male progeny, ginagamit ito sa catfish culture, and for the all female progeny, usually sa mga salmons. Okay. May sumagot din kanina ng hybridization. So hybridization is the act of breeding between two different species or genera, which ordinarily are not interbreeding. So for example here, Oreochromis niloticus kinross with Oreochromis aureus, meron siyang na-produce na hybrid. So this hybridization class usually happen, di ba, kasi sa natural spawning in the environment, sa water, nagpo-produce lang naman ng milk, nagpo-produce lang naman nung egg, external fertilization. So for example, what happens if merong different species na mix dyan, hindi siya mapipigilan na merong fertilization na mangyari. However, sabi dito, to prevent the permanent mixing of genes from these two species, yung result niya is sterile. For example, ito si hybrid 1, hybrid 2, and hybrid 3 are sterile. Kaya masasabi kayo, di ma'am, for example, nag-cross si tilapia, tapos si bangus, di merong bagong species. If ever mangyari man yan, walang tawag dito, walang pwedeng maging progeny or anak yung cross, yung hybrid ni milkfish and tilapia, it's because it results to sterility. Tandaan nyo ha, yung product usually ng mga nag-undergo ng hybridization are sterile. Okay, for the hormonal sex reversal, I think all of you are familiar with this kasi nagawa nyo na to siguro sa laboratory ninyo, sex reversal. It is the administering of exogenous steroids or substances that are not natural to the fish during the gonadal development period to override, to class sa override the expression of the genotypically determined sex. So genotypically, for example, sa tilapia, female siya. However, may pinakain ka sa kanya na lumabas ito sa board exam, lagi itong nalabas. This is the 17-alpha methyl testosterone or just MT. This is the hormone fed to tilapia larvae in fish farms with the purpose of inducing sex reversal. So why do we have to do sex reversal? Of course, this is also to promote growth and stop yung overpopulation of ponds because of spawning ng mga female when yung kanilang eggs na fertilize ng mga male. Okay, so tandaan ha, this is 17-alpha methyl testosterone. So usually, minimix siya sa feeds tapos pinapakain doon sa tilapia larvae. Okay, question number 6. The process of transferring a certain preferable traits from one species into another species, in this case, fish. So what is the answer? A, transgene, B, transgenesis, C, androgenesis, D, gynogenesis. The process. Okay, the process. May mga sumagot ng letter B, D yung mga sagot. How about the others? Letter B, okay. Most of you answered letter B. But ang very good ng mga students na ito. So, ibig sabihin, nagagaling yung mga nagtuturo sa inyo about genetics and biotechnology. So, the correct answer is... wala, hindi ko ito nalagyan ng sagot. Baka na-delete ko siya. Okay, the correct answer is transgenesis. Okay, very good. Okay, again, may tanong ako, bakit transgenesis sagot nyo? Bakit hindi transgene? Kasi parang magkapareho lang naman sila ng term. Raise your hand if you want to answer. Sige na class, pwede kayong sumagot, huwag kayong mahiya. Okay, sumagot dito si Honey from MSU na awan. Sabi niya, the term process, very good, that is correct. It really, tawag dito, yung pagbabasa ng sagot, kasi pwede mo siyang sabihin lang, transgene. However, meron ditong term na process. Okay, so the correct answer here is transgenesis. Ay, ayan pala siya. Meron pala, ayaw lang kaninang mag-next. Okay. So transgenesis is the process of transferring an exogenous DNA or yung exogenous DNA segment na yon is the transgene. Okay? Transgene to an animal so that it is able to pass the transgene on to all of the offspring. So ang tawag naman doon sa isda na merong transgene, meron siyang in-insert na transgene doon is transgenic fish. Okay, so this is the fish that has an incorporated engineered DNA into its genes. So ayan siya. Transgenesis and transgene and transgenic fish. Now, pwede naman siyang tawag dito, pwede siyang tawagin transgenic fish or genetically modified fish. Ganun lang din naman. Genetically engineered fish. Same lang, same terms. So question number 6, an animal or plant that has been created through genetic engineering. Letter A, transgene. Letter B, GMO or genetically modified organism. Letter C, hybrid. D, all of the above. What is the answer? Okay, may mga sumagot ng letter B. How about the others? May sumagot ng letter D, all of the above. Transgene. GMO, hybrid. Okay, so what is the correct answer? The correct answer is GMO. Bakit hindi pwedeng transgene? Ano nga ulit yung definition ng transgene? That is the exogenous DNA which is being inserted to the donor fish. Okay, so the correct answer is GMO. Okay, so ito yung sabi ko kanina, this is the GMO. Meron na tayong GMO fish class ha. So this is the regular salmon. However, it was genetically engineered to look like this. So this is the Aqua Advantage salmon. And did you know na itong fillet ni Aqua Advantage salmon is already sold in the US? So paano ba naging GMO itong si Aqua Advantage salmon? So gene from Chinook salmon and a gene from ocean pout, okay, were inserted to Atlantic salmon, and then it resulted into a genetically modified salmon. Ibig sabihin, class, genes which conserve, which will be beneficial, for example, sa isang particular trait na tina-target mo, kukunin yun sa isang source. So basically, this is the transgene, this is the transgene, this will be inserted here sa fertilized egg and it will be hatched into a GM salmon, genetically modified salmon. So look at this. So I have a question. Tingin niyo, merong magandang naidudulot ito si biotechnology? Is this good? Small salmon and then we have this big salmon, thrice its size. Pwede kayong mag-comment ng yes or no sa chat. Okay, may sumagot ng yes. How about the others? Anong nafe-feel ninyo? Yes? Yung iba neutral, wala? Okay, yes. For market and consumption, yes. Okay, so yes. Okay. However, there's some people din naman and we have to respect their tawag dito, views and rights about these GM products. Some of them, even may MS students, kasi sinasabihan ko na sila ng mga magagandang naidulot ng Fisheries Biotechnology. Some of them are still skeptical. Okay. Next. Are you familiar with this class? Kilala niyo sila? Glowfish. Si Glowfish. Yes. How about the others? Nag-comment si Aris. Estudyante ko. How about from other universities? Are you familiar with this? No. Okay. Hindi kayo familiar? Yes? No. So, hati. May mga familiar, merong hindi familiar. Ito ha. Glowfish. O, diba? This is the first genetically modified pet. Diba si zebrafish, itong si Danio rerio, this is an ornamental fish. Basically, pet siya. So, scientists from Taiwan, correct, tama ba? Scientists from Taiwan, kasi they want to improve the appearance of zebrafish. So, meron silang in-insert dito na gene, the transgene, in order to make this zebrafish glow. So, it resulted into a patented product na Glowfish. Tandaan ha? Yes, pet fish ito. Yes, pet fish. Rafael, pet fish siya. Glowfish is the first genetically modified pet fish. Kasi ito, ano siya eh, pet fish or ornamental fish kasi inaalagaan siya for the leisure of humans. So, meron siyang iba't ibang varieties. Ayan siya, ito yung mga kulay niya, starfire red, cosmic blue, electric green, and so on. So, nakakatuwa siya. So, a gene from another organism, for example, yung mga glow in the dark, yung nagpo-produce ng bioluminescence, yung gene that codes for bioluminescence, in-insert dito. Kaya yan, kita nyo, glow in the dark siya. Okay. Next, ito. Ayan, si transgenic growth enhanced tilapia. Meron na pala. Okay. So, this is the normal tilapia. And ito naman yung transgenic tilapia. Okay, so nakita nyo, double the size. And another example is ito din, transgenic growth enhanced loach. So ito yung wild type, sinabi natin, ito yung sa natural environment, and then ito naman yung na-improve na. So as you can see talaga, maganda rin naman talaga yung effect ng mga, yung result pala ng these genetically modified organisms. Okay. So these are the current and potential applications of transgenic fish. Lumabas ito sa board exam class, itong term na transgenic fish. So tandaan nyo yung kanyang definition ha, transgenic fish. So ito, the following: growth enhancement, freeze resistance, and cold tolerance, especially those fish from the temperate countries. They have genes inserted in order for them na makatolerate ng cold. Kasi sometimes, di ba, yung iba, if in a cold environment, they cannot really spawn. Salinity tolerance, siguro naririnig na ninyo, may mga tilapia daw someday na pwede nang i-breed sa seawater. Disease resistance, metabolic modification, improved product for the consumer. Of course, wild type, maliit lang yung fillet niya. Yung mga improved fish, malalaki yung fillet. Fish farming, okay? Fish farming, ibig sabihin dito, we are getting potential bioactive compounds which can be used in the pharmaceuticals. Kaya tinawag siyang fish farming. Pharmaceutical products from fishery products. Okay, next question. Question number 7. What is the product of the world's first selective breeding program for tropical fish? O ito naman, is si Glowfish, first genetically modified pet. Ano naman? What is the product of the world's first selective breeding program for tropical fish? Letter A, gift or the genetically improved farm tilapia. B, glowfish. C, get excel. D, milkfish. Okay, how about the others? May mas sumagot, letter A, A. Yung iba, anong sagot ninyo? A? Okay, lahat kayo sumagot ng letter A. Very good ng mga estudyante. Okay, baka lumabas to sa board exam ha. It is gift. Now, gift is not a GMO. Again, emphasize gift is not a GMO. It is a product of selective breeding program. Kaya tinawag siya class na genetically improved, not genetically modified. Whereas naman ito si Clawfish, it is a GMO. Si Get Excel, it is a derived strain from gift. And then milkfish. Milkfish lang naman. Wala naman ako sinabing iba dyan. So the correct answer is letter A. Now, when we say selective breeding, do not confuse selective breeding with the genetically modified organism. When we say selective breeding, we are choosing the best characteristics from two parents, okay? And then yung, for example, meron silang offspring, pipiliin din natin yung pinaka-best doon, and then yun yung i-breed. Okay? So selective, we select what is best and then we will continuously breed it. Now, yung pinakaiba naman class, for the GMO, targeted yung gene, so mabilis siya. For example, you can produce agad-agad nung isang genetically modified organism. Hindi naman sinabing agad-agad class ha, but when you compare with selective breeding, it took years. Years talaga class ito. For example, sa gift, more than 10 years para ma-produce siya. It's because it is the product of a careful and time-consuming and laborious selective breeding process. Okay. So, next. Question number 8. The process by which a progeny, baka maka-confuse kayo dito sa progeny, biglang sabihin nito class, anak, or the tawag dito, yun anak, the second filial generation, for example, is produced by the male parent with no genetic contribution from the female. Letter A, gynogenesis. B, androgenesis. C, parthenogenesis. D, triploidy. What is the answer? May sumagot ng B, may sumagot ng letter C. How about the others? Ano yung sagot ninyo? Okay. So most of you answered letter B. May sumagot ng letter C. Okay. Sa sumagot ng letter B, ano yung inyong basis? Why you have answered letter B? Bakit androgenesis yung sinagot ninyo? Okay, anong term yung nakita ninyo dyan? Male parent, andro is male, okay, gyno is female. Okay, so the correct answer here is androgenesis, letter B. So androgenesis, napoproduce dito male, okay, and then sinabi na no genetic contribution from the female. So andro, male. Androgenesis, male. Okay, ayan. So when we say, may mga sumagot din, maraming sumagot ng parthenogenesis. Okay, androgenesis, alam na natin, male lahat ng offspring, no genetic contribution from the female. Gynogenesis is the process of animal development with exclusive maternal inheritance. Parthenogenesis, natural form of asexual reproduction in which growth and development of embryos occur without fertilization by sperm. So what can you see here, class? Gynogenesis and parthenogenesis are almost pareho sila. Okay, so gynogenesis is a form of parthenogenesis. Okay, so ayan siya. Next, we have this question. Now, question number 9. The process of transferring genetic materials into a living cell using glass micropipettes or metal microinjection needles. Letter A, genetic engineering. B, electroporation. C, gene gun. D, microinjection. What is the answer? May sumagot ng letter D, microinjection. May sumagot ng letter B, electroporation. May sumagot ng letter A, genetic engineering. Okay. So, marami yung sumagot ng letter D. So, the correct answer is microinjection. O ngayon, bakit kayo sumagot ng microinjection? Anong term yung nakita nyo dyan? Sige lang, you can type. Anong term meron? Meron dyan sa sa question. Bakit microinjection yung sagot ninyo? Yes, correct. Microinjection needles. Again, sabi ko 'di ba? Class, yung sagot mismo nandoon na sa question. Okay? Microinjection needles, obviously the process is called microinjection. Okay. When we say microinjection, class, ito yung itsura niya. Commonly used method for introducing exogenous material into the oocytes, sa egg, and embryos of various species. So, yun siya. Ito yung injection. Ayan siya. Ito naman, class, electroporation, electric. Malalaman nyo, class, na electroporation yung sagot kasi merong sinasabi dyan na short electric pulses. Okay, short electric pulses. Parang in-electrify yung tawag dito, yung oocyte in order to deliver the transgene. So yan siya, electroporation, tandaan nyo lang, short electric pulses. Okay? Diyan na agad yung sagot. Pag sa pa lang, tanong na lang, ugaliin nating magbasaan, mag-analyze ng mabuti. Next question. Tayo naman next. Ayan. Ba't kami ganito na to? Okay. Question number 10. A process that preserves organelles, cells, tissues, or any other biological constructs by cooling the samples to very low temperatures. A. Gene bank. B. Cold blast. C. Freezing. D. Cryopreservation. What is the answer? May sumagot ng letter D, may sumagot ng letter B. Okay, so most of you answered letter D. May sumagot ng letter A, gene bank. Now, for those who answered letter D, kasi marami kayo, ano yung basis nyo why you have answered letter D? Sige, type nyo lang. I-type nyo lang kung ano, bakit. Bakit letter D yung sagot ninyo? Meron namang sumagot na letter B. Siguro kasi cooling the sample, kaya cold blast. How about the letter D? Process. Cold blast and freezing is also a process. Preserves organelles. Both of these can still preserve. Gene bank is also a form of preserving. Ano yung term? Cryo is equals to cooling. Okay, cryo. Okay. Anong ditong term nakikita ninyo sa question? Why you have come up with the term cryo-cooling, cryoprotectants? Freezing is also a preservation. Ayan. Okay. So, pwede din naman na tawag dito na sagot itong ibang ito, pero syempre, multiple choice class. What is the best answer? Best. Preserve. It can preserve, and then you put the samples in a very low temperature. Pag sinabing low temperature, hindi lang yan freezing. It is cryopreservation. You are using cryoprotectants. Okay? So, ito siya. Ba't kaya dyan yan napunta? Hindi ko na-transfer. So, ayan, cryopreservation. Okay, so, gene banks are a type of biorepository that preserves a genetic material. Tama din naman. Gene banks, they also preserve. However, they are a biorepository. Ibig sabihin, it is a place where you put mga DNA constructs, mga gene construct, any material, mga cells, organelles. Here, ito yung itsura niya. Okay? And cryopreservation, usually ginagamit dito are liquid nitrogen. This is used to preserve mga, ayan, ito, preserve this following biological matters. Yan, so cryopreservation, tandaan ninyo ha, cryopreservation. Next question. Which of the following is not a role of biotechnology in conservation of aquatic resources? Letter A. Improve the methodologies for defining species, stocks, and population. B. Understand the molecular basis of gene regulation, expression, and sex determination. C. Improve feed conversion ratio of fish species. D. Cryopreservation of gametes and embryos. What is the correct answer? Okay, divided yung sagot ng klase. Sige, hintayin ko pa yung sagot ng iba. Sagot pa. Okay. Sumagot ng C, may sumagot ng D, mayroon sumagot ng A. Most of you answered letter C, may sumagot ng letter B. Okay, sige. Tanungin ko si Annie. Annie, bakit sagot mo letter C?
Kasi po di ba pag FCR sa aquaculture siya, hindi naman po aquatic resources.
Okay, hindi naman siya aquatic resource. Nagkano din naman tayo, di ba, na aquatic resource? I mean, kinukulture din namin sila. Pero ano yung term dito? Ano yung pinaka-term? Bakit sa letter C? Okay, so the correct answer is letter... Bakit tayo nung mag-next? Ay, nako. Ayan, the correct answer is letter C. Now, look, again ha, be very careful in reading the question. Not a role, not a role, hindi role ni biotechnology in conservation of aquatic resources. The term here is ano ba? Conservation. Conservation, we have to conserve. So ano yun? Letter C. Improve feed conversion ratio of fish species. Tama din naman kasi more on aquaculture ito. But the rest here, this is more on conservation. Example dito, improve the methodologies for defining species, stocks, and population. Okay, so this is used in conservation. Understand the molecular basis of gene regulation, expression, and sex determination. This is also in conservation. Kasi dito rin nalalaman yung tinatawag nating genetic diversity and of course, cryopreservation of gametes and embryos. Yun talaga, conservation talaga siya. So learn how to analyze, learn how to read ha. Although very confusing siya, pero meron talaga siyang isa na hindi yun yung sagot. Okay. So, these molecular tools are done to do the following. So, basahin nyo na lang yan siya. Ayan. Actually, mention na din kanina doon sa question. So, ito siya. Ang daming mga uses si biotechnology in conservation. It is used. Okay, so, next question. Which aquatic species does green fluorescent protein or GFP is derived from? Now, for the information of everyone, GFP is used as a protein marker sa mga cancer cells. Okay, so what is your answer? Letter A, Thermus aquaticus. B, Thermococcus fumiculans. C, Pandalus borealis. D, Echuroya victoria. Ano yung sagot? Si Ma'am Nora, nakikisagot din. Go, Ma'am. Ano? Yung sagot ninyo, may mga sumagot ng C, mayroong sumagot ng letter B. How about the others? Mayroong sumagot ng letter D? D? Okay. So what is the correct answer? The correct answer is Echuroya victoria. Okay, sino ba yan si Echuroya victoria na yan? Ito siya. Echuroya victoria is, sandali, hindi ko dito nilagay. Ano bang tawag dito sa ano na to, class? This is a, sandali. Class, help me. Nakalimutan.
Jellyfish po.
Parang jellyfish siya.
Jellyfish.
Ayan, oo. So, ayan siya. Ito yung source niya. Hanapin nyo na lang yung English name nito. Sorry, nakalimutan ko siyang ilagay talaga. Okay, so hanapin nyo na yung English name kasi baka, for example, yung lumabas, English name, ganon. So it is better talaga na alam nyo si English name, alam nyo si scientific name. Now, si Thermus aquaticus, yung sumagot ng letter A, dito kinukuha si Taq polymerase, si Taq DNA polymerase, which is used in PCR. So this is not a bacteria, si Thermus aquaticus. This is a archaean, if you remember your biology. DNA ligase, this is tawag dito. For example, sa PCR process din, ginagamit din siya, the enzyme, DNA ligase, galing din siya sa archaea na Thermococcus fumiculans, and then shrimp alkaline phosphatase, Pandalus borealis, ito naman, DNA polymerase, which is also used pag nag-PCR reactions tayo from Pyrococcus furiosus. So, ayan siya. So, bakit ito nilagay? It's because I want to emphasize that our aquatic environment is very rich in materials or resources that are very beneficial in medicine, very beneficial in other fields and in other studies. So imagine, di ba, this protein from this jellyfish na to, ginagamit ito for cancer research. Okay, next question. In what aquatic species does ara A or virabitin derive from? Okay. Ano yung sagot ninyo? A, marine sponge, Tithia crypta, B, marine sponge, Cryptotheca crypta, C, Cephalosporium species, D, dinoflagellate. What is your answer? Okay, may sumagot ng letter A, may sumagot ng letter B. How about the others? May sumagot ng letter D. Hati yung inyong mga sagot. Sige, sagot pa kayo. Hintayin ko yung mga sagot ninyo. Letter D, B, A, hati kayo lahat. RE, very important ito kasi tawag dito, this is also a compound that is used to treat diseases. So may mga sumagot ng letter A. Most of you answered letter A. So the correct answer is marine sponge, Tithia crypta. Now this is the list. Ayan, si RA. Antiviral siya. This is used for against viruses. Ito naman si RC. It is anti-cancer. So, tingnan nyo na lang. Ayan, these are just some examples ng mga products which are used in the market na ngayon, and these are the sources. Okay, ito yung mga sources niya. Cephalosporins, ewan ko lang kung nareseta na to sa inyo, but it is an antibiotic from a marine fungi. Ayan. Okay, next question. Ito, I don't know if you're in your aquaculture na ano ninyo to, natatandaan ninyo ito. The use of aggregates of bacteria, algae, or protozoa held together in a matrix along with particulate organic matter for the purpose of improving water quality, waste treatment, and disease prevention intensive aquaculture system. A, biofloc technology. B, fermentation. C, microbial enzymes. D, single cell ingredients. What is the answer? Okay, mga sagot na. All of you answered letter A. Very good. Okay, very good. So, the correct answer is letter A, biofloc technology. Flocculation or aggregation. Bio, the flocculation of biological matters like bacteria, algae, or protozoa. Nakita nyo na ito class sa inyong mga aquaculture class. Sandali lang ha. Hello tayo. Okay, sige, biofloc technology, very good. So nakita niyo na ito class before, si biofloc. Alam niyo kung ano itsura nito? You can type yes or no on the chat box. Wala? Opo, okay. How about the others? Si biofloc, yes. Okay, very good. Now, biofloc is used in intensive aquaculture system. So sa mga in-upload namin na mga notes doon sa Google Classroom, makikita ninyo doon yung mga differences among intensive, extensive, semi-extensive, ganon. Okay, so this is the biofloc technology. Okay. Ayan siya, yan yung utsura niya, yan yung mga brown-brown na yan. So that is the aggregates of these biological matter. So it plays 3 major roles. Maintenance of water quality by the uptake of nitrogen compounds, generating in situ microbial protein. So it will also serve as a food sa mga kinukulture natin dyan. For example, shrimp. That's why it serves as a nutrition in order to increase the FCR. I reduce FCR pala. I mean, improve FCR, sorry. And when you improve FCR, you're also decreasing the feed costs and competition with pathogens. So for example, mayroong mga beneficial bacteria dyan, it can also kill yung mga harmful pathogens that can cause disease to your cultured species. So very good, alam niyo yung sagot, it is biofloc technology. And ito naman class, si fermentation. Have you heard about PECM? PECM, ang protein enriched copra meal. PECM. So PECM is a protein enriched copra meal. So this PECM was produced through the process of fermentation. Finerment si copra meal. Why? It's because most of the feed ingredients that we have now, especially if it is derived from plants, it has anti-nutritional factors. Okay? So these anti-nutritional factors, for example, yung mga phytates, ayon, yung mga lignin, this can, pag hindi siya natanggal, for example, nilagay mo siya sa feeds, it can affect. Okay, nawawala yung effect ng mga nutrients or ng feed na pinakain mo sa iyong cultured species. So that is the anti-nutritional factors. When kung 2018, nung kumuha ko ng board exam, lumabas ito si anti-nutritional factors. For example, saan dito yung example ng anti-nutritional factors? So read, ano-ano ba itong mga anti-nutritional factors? It's because madami din sila. Okay. So, enzymes, gumagamit tayo ng enzymes and feed ingredients. So, the purpose of that, these enzymes na to like lipase, trypsin, chymotrypsin, amylase, maltase, cellulase. Now, class, usually, ito sinasabi ko always sa mga students ko, malalaman mo that it is an enzyme because of the suffix ASE. Merong suffix na ASE. So, malalaman mo siya an enzyme. So, for lipase, this is an enzyme that breaks down lipids. Okay, amylase breaks down carbohydrates, maltase breaks down malt. So, ayun siya. Trypsin, this is also for protein, breaks down protein. So, why we use enzyme in feeds? Of course, we have to, tawag dito, remove these anti-nutritional factors. Okay, yun yung example ng mga anti-nutritional factors. Lectin, saponin, phytic acid, usually nakikita ito sa mga root crops. For example, si cassava meal, pag hindi nyo ito pinrases, for example, hindi ito dinrysisan, hindi ito nag-undergo ng fermentation, or walang nilagay na enzyme, may mga phytates dyan that can affect yung FCR ng fish. Okay, phytic acid is not degraded by heat treatment during pelleting. And yan nga yung kanyang result, it can interfere in the absorption of some nutrients. That's why the use of enzymes and fermentation is really necessary. Okay, now this is an example of fermentation, the solid state fermentation. You are using these mga waste products na ito as a substrate to grow microorganism. It's because we want good bacteria in the water and also good bacteria doon sa gut ng ating mga isda in order to promote disease resistance. So that is solid state fermentation. You are using these mga wastes as substrate. Yung substrate kung saan ka nagpapagrow ng mga microorganisms, ng mga good bacteria. So now, LSPU, meron kaming project wherein we are doing the solid state fermentation. Small scale pa lang naman, and we are using molasses, okay? Parang sugarcane din siya, sugarcane bagas, molasses as a substrate wherein we grow lactic acid bacteria. Okay, so did you know that bacteria can be also an ingredient in fish? So these are the microbial ingredients in aquafeed. So merong dalawang klase. Number one, you have the SCI or the single cell ingredients, and then the SCP or the single cell protein. So for the SCI, it includes bacteria, fungi, and other mga algae, okay, or combination of these three. Whereas naman itong si single cell protein is a bioprotein. So ito na siya, galing din siya dito kay SCI, but it is in a broad source. Okay, broad source siya. And then this was already aggregated. Okay, question number 15. What are live organisms that are intended to have health benefits when consumed or applied to the body? A, prebiotics, B, probiotics, C, postbiotic, D, symbiotic. So what is your answer? Sumagot ng A, may sumagot ng B. How about the others? Is it prebiotic, probiotic, postbiotic, or symbiotic? So most of you answered letter B. Okay. The correct answer is letter B. When we say probiotic, na inom kayo ng Yakult, probiotic is a live microorganism. So Yakult is an example lang ng food na merong probiotics. Tandaan nyo class, pag sinabi natin na prebiotic, versus probiotic, prebiotic is, okay, usually plant-derived. Galing siya sa plants. Okay? Plants. Pag probiotic naman, these are live bacterial culture. The combination of these two, tinatawag na symbiotic combination of prebiotics and probiotics. Now, para mas maintindihan ninyo, si Yakult na iniinom natin, it is a probiotic, pero alam nyo ba, meron tayong iniinom na symbiotic? Kilala nyo si Delight class? Nakainom kayo ng Delight?
Opo.
Wow. Ayun, si Delight. Delight is an example of a product na merong symbiotic. Basahin nyo mamaya kung meron kayong Delight dyan. It contains both prebiotic and probiotics. Okay? Next. Bakit we use probiotics sa aquaculture? It's because feeds, and when you put it with probiotics, it improves gut microbiota. It's because merong mga good bacteria dyan, enhance growth, improve digestion, and improve immunity. So same as din class sa atin, uminom tayo ng Yakult, it's because okay sa tiyan. Okay, di ba? So same din class with the fish. Nilalagyan din sila ng probiotics to have these health benefits to them. So ito yung mga example ng mga commercial probiotics that we can get, especially ginagamit ang probiotics sa intensive aquaculture. Okay? Next question, second to the last question na to. A technique used to rapidly increase the number of copies of specific regions of DNA. Is it A, polymerase chain reaction, B, gene probe, C, blotting, D, genetic engineering? What is the answer? Yeah, may sumagot, prevention is better than cure. Tama, tama. So you have to prevent the disease before it can spread. Okay, the correct answer is, very good, kayo lahat. The correct answer is polymerase chain reactions. This is the process to amplify or make a multiple copies of DNA. So for example, pag DNA class, it is PCR, but if you're going to analyze RNA, yun yung tinatawag na RT-PCR, which is alam na alam natin ngayon na reverse transcriptase PCR, polymerase chain reaction. So polymerase chain reaction class, this is used for the prevention, control, and management of various diseases. So this is used, naalala ninyo yung lecture natin last time about white spot syndrome virus. So sabi dito ng student, sabi ni Joshua, prevention is better than cure. That is correct. Now, this PCR is used as a prevention. Usually, in a farm ng mga shrimp, kasi di ba intensive culture sila, kumukuha lagi doon ng sample and then tinitest if baka merong presence ng virus, okay? Viral DNA or viral RNA. So, tinitingnan. I-amplify. O ibig sabihin, kung merong na-amplify, merong presence ng virus. So, it is a valuable tool na ginagamit para malaman if baka magkaroon ng outbreak ng disease. For the gene probe naman class, this is a DNA probe. Okay, so it is a fragment, DNA or RNA fragment used in genetic engineering to search for a particular gene or other DNA sequence. So DNA probe, pwede siyang isang, this is just a DNA sequence. Ayan, to determine the presence of luminous bacteria. Yes. Blotting. Okay, si blotting naman, meron itong tatlong klase, si southern, western, northern. Baka tanungin niyo ako, ma'am, bakit wala si eastern? Hindi ko rin alam. Pero yung una dito na-discover is si southern blot. Yung naka-discover sa kanya, bakit ganito yung kanyang mga pangalan, is si scientist na yung apelyido is si southern. Kaya yung the rest, pinangalan na din na western tsaka northern. Now, tandaan nyo ha, southern is used to detect DNA. Western is used to detect amino acid sequences in proteins, and si northern detect RNA. Depende na sa inyo if paano nyo ito tatandaan, pero ako usually, sinasaulo ko na lang talaga eh. Wala akong specific na mnemonics dito or technique para matandaan ito. Basta I know, ayan, southern blot, si DNA, western blot, si amino acid, northern blot, si RNA. So, hanapan nyo na lang na way on how you will remember this. And the last question is, which of the following is used to prevent diseases in fish? A, vaccine, B, antibiotics, C, fish immunization, D, all of the above. What is the answer? D, may sumagot ng letter A. How about the others? D. Okay. Most of you answered letter D, all of the above. Ay, which of the following is used to prevent? Mali class, mali ito, sorry. The correct answer here is all of the above, hindi vaccine ha, hindi lang vaccine. Hindi ko lang to napalitan, sorry. But the correct answer here is all of the above. Fish immunization and vaccination or vaccine, pareho lang yan. And also same with antibiotics, it is also used to prevent diseases in fish. So when we say vaccine, okay, this is a biological preparation that provides immunity to the fish, for example. So there's 3 methods to administer vaccine. Immersion, i-immerse mo doon yung fish on the vaccine injection. Okay, and oral, which is done through mixing doon sa feeds. And the process wherein you put vaccine, of course, is vaccination. Lahat tayo dito familiar. So tandaan, usually ngayon, yung ginagamit na vaccine sa fish is in the form of DNA vaccine. Now for antibiotics, yes, ginagamit ito. This is used to kill or inhibit the growth of bacteria. However, one drawback of using antibiotics is ina-abuse. For example, merong sakit yung isda, lalagyan lang ng antibiotics. So what will happen? This will lead to antimicrobial resistance. AMR or antimicrobial resistance happens when the bacteria, when the antibiotics has no longer effect on the target bacteria. Kahit maglagay ka man dyan ng matataas na dosage ng antibiotics, it has no longer an effect to the bacteria. It's because the bacteria has already evolved to resist these antibiotics. So, ayun siya. Yan yung isang drawback. Kaya ngayon, yung ginagamit is vaccine. Okay? Yung ginagamit ngayon is vaccine or naglalagay na lang ng symbiotics or pwede ding probiotics sa feeds. So yun yung purpose ng aming project dito sa LSPU. So for 4 years now, 2 projects yun. Because ito nga gustong pigilan itong AMR na to. So we have developed a symbiotic which when given to a fish, it really improved their immunity against diseases. So ayan siya. Ito kasi lahat na yata ng mga speakers meron na sinabi sa inyong mga tips. Alam ko marami na kayong nakuha tips. Ito akin lang. Ito yung aking motto. Sinulat ko to sa notebook ko when I was reviewing sa notes ko. Sabi ko, I am like a salmon. Okay? Si salmon, di ba, anadromous, galing sa seawater, tapos pupunta upstream sa fresh water in order to spawn. So ito nilalagay ko, swim upstream and reach the top. Now, ano yung na-observe ninyo kay salmon? Diba, before niya ma-reach yung upstream, ang daming struggle niyang dadaanan muna. So same din, when we are reviewing for the board exam, ang dami nating mas struggle na makukuha along the way. So si salmon, in order to reach the top, anong ginagawa niya? Ang dami niyang effort na ginagawa. Too much energy. Effort is being expended. So ganito din sa atin. In order to reach the top, and in order for us to swim, merong sumusulat sa aking screen. Okay? In order to reach the top, okay, we should also put more effort. Ibig sabihin, mag-effort pa tayo on how we study. Okay, hindi lang yung kung ano lang pinrovide sa atin, yun lang. Mag-effort din tayo sa sarili natin. So hopefully, yung i-reach nyo, hindi lang yung passing score, you have to reach top 1. And sana, sa parating na October, marami kaming makita ng mga board passers and hopefully, mga top passers sa parating na board exam. So thank you so much. Thank you for listening. I hope may mga napag-aralan kayo. So this will be uploaded on the Google Classroom. Thank you so much. Okay, so if you have any questions, pwede na kayong magtanong. Ma'am Pat, pwede na pong mag-stop recording.
Okay po, ma'am. Thank you.
Okay, attendance and then evaluation.